too. Okay, so what do we do with the cytoskeleton? Now, the main part of the cytoskeleton that gets affected by the extracellular matrix is made of what we call the actin microfilaments. Now, all eukaryotic cells are loaded up with microfilaments. They're made of this protein called actin. By the way, fairly soon I'll post up a thing on Blackboard, which we probably won't get to in class, but if you want to review the cytoskeleton for your own uses or for future classes, they'll be there fairly soon. I just have to do some editing on it. Okay, well, the microfilaments, they're thin filaments made of this protein called actin. The thin filaments of muscle are actin microfilaments. It's a specialized arrangement of microfilaments that we find in muscle cells. But all cells have them. Most of the microfilaments are close to the cell surface, just underneath the membrane. They play roles in moving things around inside the cell, and a lot of roles in cell movement. When cells crawl around, think of amoeba and stuff like that. They're relying mostly on their microfilament cytoskeleton. There's a whole bunch of proteins that regulate this stuff, cause these filaments to form, cause filaments to break down, assembles them together in networks and stuff like this. A whole bunch of regulation. Well, we're going to work through this. Now, what's going to happen is we can bind to several proteins. There are a cascade of proteins, these integrins. And two of the most important ones that act as adapter proteins between the integrins and the microfilament cytoskeleton, one called vinculin, which binds the integrin, and which binds it as something called talin. Okay, now, the talin will then bind to the end of a microfilament, which is a double row of these actin molecules. Okay. Additionally, so what's going to happen here is you're going to anchor this integrin, which is this membrane proteins, to this microfilament, this rigid protein fiber that plunges deep into the cytoplasm. So we're anchoring these, and when you have a whole bunch of integrins together, you can anchor a whole bunch of them into almost like a cable that plunges into the cytoplasm. And we'll see that in a little bit. That, by the way, is what we call focal adhesion. We'll see that a little bit later. Now, additionally, this is also going to activate three small monomeric G proteins. They're distant relatives of RAS, but they have nothing to do with signaling. And these three proteins, it's not a direct binding, by the way, are called RAC, RHC, RHO, RHO, and CDC42. They're all G proteins. They're all monomeric G proteins. And when these guys are activated, what they ultimately do, for a few steps, is they're going to stimulate the formation of additional microfilaments. What they will do is activate a complex of proteins called the ARP2-3 complex. And it's a series of several different proteins. The thing forms like a cylindrical rod, sort of about the same shape as this piece of chalk here. Now, what these guys do is this. Here you have a microfilament. We'll put MF. Don't read this the wrong way, folks. Okay. Now, when activated, what will happen is this ARP2-3 complex binds kind of to the side of a microfilament, and then it stimulates the addition of new actin molecules to the end, forming a new one. So now you start getting a branching network that looks kind of like this. You start out with one, and now you have a branch coming off, you have a branch coming off of that one, and more branches, and so on. And now you make a dense, three-dimensional network of the cytoskeleton. And that can be involved in cell movement and cell shape. So we're seeing the integrins are playing big roles in stimulating changes in cell shape, changes in cell movement. So they're affecting the cytoskeleton. You look like you have a question back here. Yeah?
I'm just trying to read what the G proteins
are. Oh, okay. These are three G. One's called RAC, R-A-C. The other's called RHO, R-H-O. and the other is called CDC42. That's cell division cycle, not Center for Disease Control. Anyway, okay, and they indirectly activate this ARP2/3 complex, and that stimulates the formation of new microfilaments, new cytoskeleton. And what can happen when this occurs is it can also push the cell membrane out. As you get more and more of this filament network expanding, that can actually push the cell membrane out, generating a cell extension. And that's exactly what happens here. So you have a cell over here, and now we fire all this stuff up over here, and what's going to happen is you're going to get a cell extension like a pseudopod or something. It's going to be growing outward. Whoa! And you may have them coming out multiple places. So now we got a cell that can potentially do things like move around or crawl around or what have you. All right. Now, not only are we going to stimulate these cytoskeletal changes, we're also going to anchor these cells to the extracellular matrix. And once again, it's going to require the integrins. So what we're going to form are these structures called focal contacts or focal adhesions. Remember that's what that kinase is there for? Now what's going to happen is this. We're going to scale that drawing we just had down quite a bit. Here you've got a cell membrane. Okay? Here's lots of ECM. Okay, here's some integrins. Integrin over here. It binds the ECM, and we get a microfilament attaching to it. Now we can have another set right over here, and it binds, and we have another microfilament attaching. We continue this process on, and pretty soon we have a big bunch of clustered integrins, each attached to the extracellular matrix, and each attached to a microfilament. And it forms a bundle. And it plunges deep, deep into the cytoplasm. So now you got a solid anchor of the cell to the extracellular matrix. And these structures are known as the focal contacts or focal adhesions. Now these things, once you make them, they're dynamic structures. You can make them in a matter of seconds and break them down in a matter of seconds. You can regulate some of these attachment proteins, and the integrins fall off of the microfilaments and then they just drift around in the membrane, or you can cause them to cluster together again, forming a focal contact. So we have these things. You can form these guys and you can get rid of them rapidly. And each cell that's attached and crawling along the surface like that is going to have multiple focal contacts. And that's what sticks the cell to the surface. So here we have a cell here. And we're going to have these focal contacts in several different places. Or focal adhesions, whatever you want to call it. In effect, they act as spot welds, gluing the cell to the extracellular matrix surface. But we can form and get rid of them as a cell needs. And that can lead to things like cell crawling. Now, has anybody seen either a video or a live under a microscope, we saw preserved slides of amoebas and stuff, but like a living amoeba? You see that the thing looks like kind of a droplet. You see it stretches out a cell extension called a pseudopod. And you look closely, you see material flowing into that. And then eventually, a good portion of the cell flows into that and it puts out another pseudopod, and that's how it crawls around. A lot of cells, the fibroblasts, a lot of these cells, the fibroblasts that make up cartilage, many of the cells in the immune system do the same thing. A lot of cells crawl on these extracellular matrix surfaces. Now, how do they do that? Well, it's been relatively past 10 or 20 years that we have a good understanding of that process, excuse me, on the molecular level. But why is this important? Several different things. We'll see how this works in just a few moments. First thing is this. cells can crawl around along specific pathways and settle down in specific destinations. This is very important in development. Now think of this. We were talking about these neural crest cells, right? That come off the top of the neural tube and they spread all over the place and stuff. Why do some of them migrate to where the future jaw is going to be? Others migrate to other parts of the body. How do these cells know where to go? Another case in point. The cells that will eventually be the predecessors of the sperm and egg, what we call the primordial germ cells. Those cells in our early embryonic life are not even in our bodies at all. They're in tissues located close to the embryo, but not in the embryo. And then later on, it's still early development, these cells crawl into the embryo, migrate to a specific part of the embryo, and settle down there. And later on, the gonads develop around this cluster of primordial germ cells. How do those cells know to go where the gonads are going to be and don't end up where the stomach is or where the brain is or where your toes, future toes are going to be? They have to somehow know what direction they're going. Now another thing, think of the central nervous system. When you're born, you already have a physiological and behavioral operating system installed up here. Nobody has to teach a baby how to suckle, for instance. Baby already knows how to do that. And baby will respond to different kinds of stimuli. Baby has the ability to recognize whoever its caretaker is, whether it's the mother, whether it's somebody in an orphanage, whether it's the nanny, whatever the case is. The one that gives the most care, baby will recognize and kind of attach to that person. Okay, they don't have to be taught that. Those neural pathways are already laid in before you're even born. So the question is, how do you hook up the central nervous system? Now obviously we're constantly generating new neural pathways, especially in childhood, but even in adulthood. So like hopefully after the exam, some of you folks might actually remember some of this stuff. Or we'll come back if you take biochemistry and say, oh yeah, I remember this signaling pathway or that signaling pathway. You've altered some neural pathways. So now you might remember some aspects of cell biology when it comes in handy in the future. And for many of you folks, it will, right?
Oh, yeah.
Take biochem, definitely. A lot of people say this really helps out. In future, medical school, graduate school, whatever the case is. Okay, so you're constantly making new neural pathways. And the thing is, how does the brain wire itself up? You're talking about, several tens of billions of neurons that have to make connections with the appropriate other neurons to have this behavioral and physiological program laid down before you're even born. Well, here again, that's another kind of issue. And part of it, in all of these cases, there's other factors in the central nervous system too, but part of this cell migration is due to the extracellular matrix. And let's see how that works. Now, what's going to count is this. Each cell has a certain population of integrins. And different cells vary on what different integrins they have. There's scores, if not hundreds, of different possible integrins that cells can have. Each set of integrins is going to bind to certain extracellular matrix things, but not others. For instance, some integrins may bind to certain fiber-forming collagens, but ignore the sheet-forming collagens. Others may bind to fibronectins, but not so much to collagens, and so on. Okay, so each integrin has a preference. So we'll say integrin binding preference. Now, when an integrin comes into contact with an extracellular matrix that it likes, in other words, that it can bind to, that's going to stimulate the signaling and that's going to stimulate the cytoskeletal changes. So a cell that's in contact with the right kind of extracellular matrix, and here's a top-down view, is going to start putting out cell extensions. It's going to sprawl out, it's probably going to crawl a little bit, maybe randomly or what have you. A cell that comes into contact with an extracellular matrix surface it doesn't like, in other words, the integrins don't bind to stuff very well, you're going to have fewer or no cell extensions. And the cells aren't going to move. In fact, they'll probably round up and cringe up when that happens. And of course, if you put cells in no extracellular matrix, most of them round up, cringe up, and don't do anything except sit there staring at you. Okay, so that's part of it. The integrins are going to be stimulating cell movement. Now, let's suppose we have a situation like this. We're going to have, effectively, a compositional trail of extracellular matrix. Let's suppose, and this is just an example here, you can have any kind of example you want. Let's suppose we've got a cell that has integrins that bind strongly to fiber-forming collagens, but not to other kinds of proteins. So we could say here, so in one part of the organism, you have very low levels of it, and somewhere down the line, you have higher and higher levels, and then somewhere continuing on, the levels start dropping down. Now over here, we're going to say, let's suppose a certain type of fibronectin. And let's suppose we have dotted line, something like this. Okay. So now we have two gradients. This one represents the fiber-forming collagen. This gradient here represents a certain type of fibronectin. Now let's suppose we have a cell over here. This cell happens to have integrins that like the fiber-forming collagens. What's going to happen is this. Even the small space of a cell, 5, 10, 20 micrometers, you'll get more integrin binding at the area that the concentration is the highest. Even though it's not very much higher, it is a little bit higher. So you get more integrin binding here and less over here. Now what that means, when you bind the integrins, you're going to start sending out cell extensions, pseudopods and what have you. So what happens is once in a while we get a cell extension out over here, but more often than not we'll get cell extensions out in this direction. And cells tend to crawl, and we'll see cell crawling a little bit later, cells tend to crawl in the direction of these cell extensions. So if you've got preferential cell extensions out here, it means the cell's going to, overall, is going to tend to crawl in this direction, towards the higher concentrations of the collagens it likes. So here we have more cell extensions here because there's still more integrin binding, less over here. Cell continues to crawl. And it continues even more. Let's put a cell extension out here. So the cell can recognize that there's more of what they like in one part of the cell and less in another. And they will head in the direction of the preferred composition. Now meanwhile, there could be a cell out here that happens to like, and I'll put it as a square, cells usually aren't like that, happens to like a certain type of fibronectin. Once again, it will continue to crawl up here towards the higher concentration of fibronectin. Now what happens when you finally get up here? You're at the peak. You put out cell extensions pretty much evenly. If you happen to crawl, if you crawl a little bit in this direction, what's going to happen? There's less stimulation here, less integrin binding. You're less likely to get extensions here, more likely to get them in this direction. If you crawl down this direction, here again, you're going to tend to preferentially crawl towards the higher concentrations. So once you reach your peak concentration of the extracellular matrix you like, although you're going to move around a little bit, it's going to be kind of random, and it's going to be centered on the area of highest concentration. Kind of like, picture this, kind of like, let's suppose, sometimes happens to a conference. Let's suppose you're out with a bunch of students and colleagues and stuff, and you're at a conference and you do the pub crawl, right? Okay. Now, yeah, it happens once in a while. Now, let's suppose you overdo the pub crawl. Some of our students, and occasionally some of us, have done that on occasion. Okay, now, you're coming out. You still like good quality beer. You walk out of one bar, and you're pretty hammered, and you're staggering around, but you prefer the next bar that has the beer. So you're kind of staggering around. but you're going to be preferentially moving, if you, well, you still can, in the direction of that bar, right?
Yeah.
Yes, and then when you finally get to the highest sorry, the highest concentration, the bar itself, the highest concentration of beer, you kind of settle down in there. And then you have some more beers, and then you pass out, and your colleagues carry you back to your hotel room.
How many times has this happened?
Well, it's happened to students. I remember one time we had a conference in New Orleans, and the very first night, one of our graduating seniors was fortunately a very petite woman, had to be carried back by a couple of her much larger friends, which is a good thing, all the way from the French Quarter, half a mile to the hotel room. She was not in good shape the next morning. Okay, but anyway, you know, you get this kind of thing where you're moving around kind of randomly, but your general direction is towards the source of the highest concentration of what you like. Now, the neat thing about these extracellular matrix trails is you can have multiple trails in the same region. In this case, a cell that likes these fiber-forming collagens moves up here and settles down here. A cell that likes a particular type of fibronectin can move the same thing, but they settle in a different place. And this is how cells, at least a major factor, not the only thing in all cases, but this is how cells can migrate from one part of the embryo to the next and know where to settle down. They settle down in the region that has the extracellular matrix they like the most. It gives them the most integrin binding and integrin stimulation. Now, one last aspect of this, just taking a look at cell crawling in general. Now, I'm going to refer you to a YouTube video taped a few years ago, except that sometimes this demonstration tears buttons off my shirt, so it's been taped. So if you take a look at that one too, it is kind of funny. But at any rate, cell crawling, how do cells crawl? Okay. All right. Well, here again, we're going to have extracellular matrix. We've got a cell here. Now let's suppose we've got a gradient of what the cell likes this direction. You're going to get more integrins binding here, and you're going to get preferential cell extensions. So here's a cell extension that's formed. The microfilaments are generated, and the microfilaments expand. They get longer and longer. Okay, as the cell crawls, we have these focal contacts here, or focal adhesions, we'll call FA here. These anchor the cell to the extracellular matrix. The cell puts out an extension. And what's going to happen is we can form new focal contacts here, and at the same time, we can get rid of the ones here in the tail end of the cell. Remember, we can form and disrupt focal contacts in a matter of seconds. So we've got that. The cell has moved in this direction, made new focal contacts, got rid of old ones. Now the cytoskeleton, mostly the microfilaments again, are going to do two things. One is you're going to pull material in here, and the other thing is you're going to squeeze the cell at the tail end, kind of like squeezing a water balloon. You know, if you squeeze a water balloon at one end, the water goes into the next end. So a combination of squeezing and pulling is going to move the cytoplasm towards this cell extension, which starts swelling up. So it gets bigger and bigger and bigger. And this end kind of starts to retract. And then we repeat the process. New cell extension, new focal contact, and stuff like that. Now, like I say, on YouTube, there is, a couple of years ago, that was recorded of me trying to imitate a crawling cell myself. It's pretty funny, and it illustrates the thing, too. Me trying to imitate a crawling cell on a bunch of these desks and stuff is kind of interesting, but it's worth watching. It's kind of funny and stuff, and it does illustrate the principles maybe a little bit more dramatically than they did today. So I recommend you want to pick up a few things on cell crawling. Actually, just help on tests.
I think everyone at JSU has watched that video.
What video is it?
What?
What video is it?
Oh, it's, I think, so, like, what was it?
like humping a desk.
Yeah, there's something like humping a desk.
You're humping a desk.
Well, that's what it looked like, I guess, yes. Oh, that would just represent an extracellular matrix that this particular cell will group. Low levels here, high levels here. So cells going to move in that direction preferentially. Okay. I used to do that, I used to do that fairly frequently, that demonstration fairly frequently. One, sometimes it does get you a little tired. And two, I've torn numerous buttons off my shirt in the process. I'm kind of like, maybe getting a little bit too old that frequently. Now that's up there for posterity. But gotten a lot of comments about it, including many people that are not from JSU. All right. It's a little more dynamic than some of the lectures we have.